Okay, guys, so this is going to be a little awkward because I'm actually recording this while I'm on hall duty. So, you can see where I am because the business ed teachers are behind me. But here we go. I got to get this done so that you guys can do it for homework. So, blood typing. So, there are lots of different ways to classify blood, but the ABO blood typing system is the most commonly used, and it's the one that's most commonly known. So, you guys have heard about somebody who's like A positive or B negative or something like that. So there are 4 different blood types in the ABO system. You could be type A, you could be type B, you could be type AB, or you could be type O. The type of blood that you have depends on different antigens that are present on your red blood cells. So I have a red blood cell right here, and it has an antigen on it. And an antigen is just a protein. So all it is it's a protein on the surface of the cell that acts like a name tag. So type A red blood cells have type A antigens. Type B red blood cells have type B antigens. Type AB blood cells have both type A and B antigens on them. And type O blood cells are naked. They have no antigens at all on them. Okay? So here's another graphic that you can look at. So the A antigen looks like this. So a blood type A red blood cell only has those A antigens on the outside of it. A type B antigen looks like this, and this is not in real life. This is just for this diagram. So blood type B blood cells have these little antigens on them. Notice that this guy has type A and type B antigens on the outside, and blood type O has nothing. Ignore this part about universal recipient and universal donor. So remember that you have antibodies in your blood. Antibodies are shaped like this. Antibodies are just little proteins, and they are there to help bind to antigens. So antibodies are just proteins, and they're produced by white blood cells, and their job is to attack invaders. And the way that an antibody knows which cells to attack and which ones to leave alone is by the identifying marker on the surface of the cell. So if we're looking at these cells with these antigens on them, only antibodies specific to these antigens are going to be able to bind. So this guy, this antibody is going to come in and it's going to go, I can't bind with you. I can't bind with you. But if this guy comes along, this guy's going to go, oh, we're bonded. So nice. And this guy could come along and this guy could go, I can't bond with you. But it could go, oh, we're bonded. So nice and so cool. So you have Y-shaped antibodies, and then the antibody has a very specific binding region on it that can only match up to a very specific antigen. Okay? So what happens then is that you might have those particular antibodies that meet up with cells that have the right antigens. And if that happens, then you can have the blood agglutinate, which means it clumps up, because every one of these antibodies has 2 binding sites on it, which means every one of these antibodies could actually bind onto 2 different cells. So then if we had more antigens over here and more antibodies and more cells with antigens and antibodies, you could end up with a whole clumped up group of cells. Okay? Agglutinating is just the fancy science word for clumping up. Agglutinate just means clump up. So you can see here, this antibody has grabbed onto this cell, and it's holding them together. And at the same time, this antibody has grabbed onto this cell, and is holding them together. This antibody is holding onto this cell, and is holding them together. This guy is sort of bridging the gap between these cells. So you have this whole clumped up thing going on. So do we want to have type A blood having anti-A antibodies in it? No, because if we had type A blood with type A antibodies in it, then our type A or our anti-A antibodies would always be clumping up our blood. And if your blood is clumped up, it cannot flow through your system. So what kind of antibodies is type A blood going to have? Hopefully you said it's going to have type B antibodies, because this guy can't bond with this guy. There's no, so you won't get any clumping going on, okay? So if we're looking at type A blood, it's going to have an A antigen on it, and it's only going to have anti-B antibodies. Yes, it's going to have anti-B antibodies on it, okay? If we have type B blood, if we have type B blood, it's going to have B antigens on it, and it's going to have anti-A antibodies on it, in the blood, not on it, in the blood, okay? If we have a type AB blood cell, then we have type A antigens, type B antibodies, and we don't want to have either one of these in there because either one of these is going to cause problems. So we just have no antibodies if we have type AB blood. It doesn't mean no antibodies at all ever. It just means no blood type A antibodies. If you have type O blood, then you are going to have both of these antibodies in your blood because there's nothing for it to do to clump up, okay? So you got to start by looking at a blood cell and saying, okay, what kind of antigen does that have on the blood cell? Whatever type of antigen it has on the blood cell, that tells you then what kind of antibodies it's okay to have in the blood, okay? So when we determine blood type, what we do is we use different sera, and a sera, that's just a plural of serum. So we just use these different serums. So anti-A serum is just a bottle. It's just a container that has anti-A antibodies in it. So it's just going to be a bottle of these little dudes, okay? Which means if I have a type A blood and I put this in it, type A blood is going to clump up because the A antigens on this are going to, and this are going to clump up together. Now, the same thing is going to happen with AB blood because AB blood also has A antigens. So if I take a serum that has these guys in it, it will make type A blood clump and it will make type AB blood clump, okay? But if we're talking about just type B, there are no A antigens, so there's nothing for this guy to grab onto. If we're talking type O blood, there's no antigen, so there's nothing for this guy to grab onto. Anti-B serum is going to have only anti-B antibodies. That means it's only going to clump if we put it into blood that has B antigens. So that's either going to be our type B blood cells or our type AB blood cells, okay? Type A blood will only clump with anti-A serum. Type B blood will only clump with anti-B serum. Type AB blood is going to clump with both of them. Type O blood is not going to clump up at all. You can put whatever serum in there you want, type A or type B, anti-A or anti-B, you're not going to get any kind of clumping, okay? So when we start to look at population frequencies, depending on what source you look at, and I know your book has slightly different information, depending on what source you look at, you're going to get approximately these values. So in our population, about 40% of our population has type A blood. About 10% of our population has type B blood. About 5% of the population has type AB blood. That's me, I'm super rare. And about 45% of our population has type O blood. Okay, if you haven't written those down yet, make sure you write them down because you are going to need those as we carry on. So RH factor. So this is that positive or negative thing that people talk about. RH factor is just a different antigen that may or may not be found on a person's blood cells. So it's just a whole nother protein that's either there or it isn't there. So that's this blue dude. So this would be a type, I've lost track of my colors, A. This would be a type A positive blood cell, okay? So Rh positive blood has the antigen, which means it will not have these antibodies, okay? It's got the antigen, so no Rh antibodies. But Rh negative blood does not have the antigen, so it will have the antibodies. It's going to have the anti-Rh antibodies because there's nothing for it to bind to and nothing to clump things up, okay? So Rh positive, Rh negative. It's just a whole nother thing. Doesn't matter in terms of whether we're talking about A blood or B blood or O blood. So actually all those blood types, you can have A positive blood, you can have A negative blood. You can have B positive, you can have B negative. You can have O positive, you can have O negative. You can have AB positive, you can have AB negative. We've actually got all of those other blood types. So we've got a whole different factor there that we can look at that's called the rhesus factor. It's named after monkeys, rhesus monkeys. They're chocolatey and peanut buttery. No, I don't know why they're called rhesus monkeys. It's spelled differently. So RH frequencies, entirely unrelated to the ABO frequencies. About 85% of people are RH positive, and about 15% of people are RH negative. Again, 85% of people are RH positive, 15% of people are RH negative. If you haven't written that down yet, write it down now. Ugh, my answer came up first. Okay, so if you want to figure out the population frequency for combined blood type, just multiply the two probabilities. So the population frequency for type A positive blood, well, the population frequency for type A is 40%. But remember, when we're going to do math with a percent, we have to do that whole decimal thing. So 40% turns into 0.4. So 2 places to the left. I don't know which way is the left for you. 2 places to the left, 0.4. The population frequency for RH positive blood is 85%, which is 0.85. Okay, and then you just multiply them. So 0.4 times 0.85 gives you 0.34. And do the percent thing, 34%. So that means 34% of the population has type A positive blood. So if you find A positive blood at a crime scene, that means it could be this suspect you have who also has A positive blood, but it could also be another 34% of the population. So there are quite a few people with A positive blood. So let's look at AB negative blood. That's me, AB neg. Let's figure this out. So type AB blood type has a 5% population frequency, or 0.05. What's negative? Good. 15%, or 0.15. Go ahead, hit pause, and figure out what the frequency is for AB negative blood. Okay, 0.05 times 0.15 gives you 0.0075, or 0.75%. So less than 1% of the population has the same blood type as me. I'm so rare. But that also means I probably shouldn't go leave any blood behind at crime scenes, because that could cause a problem for me. So that's it. That's all I got for you. Hopefully you got this, and you'll get to work some more on this later on. Okay, bye.